Hello everyone, welcome to Rapid Revision series, wherein we'll try to complete the agriculture optional in 20 hours. Okay, today we're going to discuss about cell ultrastructure. Thanks to Amit Kumar. So this is something brief about me. Again, personality test, and I've written multiple mains and PhD from IRI. So these are some of the reviews. All the this year's toppers like Lakshmi Priya, Harini, Ramakrishna Saran, Sushri, they're all like IAS and IPS, IRS officers this time. You would have seen them. And this like IFS officers like Vishal. So, so today we will discuss about cell ultrastructure. So the major focus will be on to complete the topic exam-oriented approach. Okay. This topic, if you go to some classes or something, they may take up to 3 to 4 hours. Yes, that may take more that much time to explain. But here we will be exam oriented. Whatever is asked in exam, how much you need to remember in exams, in a very short time, we will discuss. Okay, in a very short time, maybe in 20 minutes or so, we will complete this. We will study the important things and the bullet points which you need to write to get the highest marks. You don't write, you just know many things, then you may not be able to understand. So be very specific, understand the concept. And here there is some interlinkages with the physiology portion. Okay, so those things we will study in the physiology portion. So let's start. Let's see the PYQs first. See, the question in 2021 is define cell. How is plant cell different from animal cell? Describe typical cell structurally and functionally with suitable diagram. Again, you see here, major components that constitute cell structure. Describe their function. Similar and same question in a different form, but the answer is same. Here you see, what is plant, what is cell? Enlist different organelles of plant cell with brief description on the function of mitochondria. Maximum there was something different means mitochondria. This can be written also because you're going to study the in physiology different aspects with respect to mitochondria. So based on this, you'll be able to remember. Okay, so let's go ahead. So the first topic which we should remember is about cell wall. Okay, so what is cell wall? It's a non-living and rigid structure. Okay, and it provides a definite shape to this cell. Okay, and also what it does is it provides this mechanical support. Okay, and to the tissues and organs, it provides the mechanical support and structure. So here you can see the first layer inside. You can see this is the middle lamella. This is called the middle lamella or the cementing agent. This is like a cement which builds when we build a brick, we add cement. So in this way, this is the middle lamella. Then next we have the primary cell wall. Okay, after middle lamella, we will have the primary cell wall, then the secondary cell wall, which is more thicker. This is light thicker, this is more thicker. We'll study them now. Then last we have the plasma membrane that is most inside the cell. Okay, so what you understood first is plasma lemma, then secondary, primary, and then the cementing agents, that is middle lamella. Okay, so now first let's see how this middle lamella, what are the important aspects. So the present, it is present between two adjacent cells, is composed of pectins in the form of calcium and magnesium salts. You should remember one pectin should be written when you write about middle lamella. Okay, this I'm telling you, like you see, you have 3 pages in an 15 marker. So whatever you need in that much, if you read for 10 pages, what do? You can't write all these things, but I will tell you content to write in 3 pages, all key points, and nobody can give you less than, like if you 15, some 8 or 9 marks. Okay. So it is an adhesion of the walls. Okay, it is like adhesive. Okay, of the contiguous cells, and the pectin readily hydrolyzed by pectinase and strong acids. It can be only be hydrolyzed and broken by these pectinase or some strong acids. And this is the first to originate in terms of cell division. Second structure I told you is primary cell wall. I told you this is less thicker compared to secondary cell wall. Why is it so? Because it is made up of hemicellulose. Okay, hemicellulose, it is 53%, then cellulose 30%, and 5% pectin and protein. and lipids. But here you should observe that hemicellulose is 53 and cellulose is 30%. Why am I telling it? Because we will see in the secondary cell wall the percentage difference. Okay. So this you should remember. And you should also remember this point, that meristematic cells are made up of primary cell wall only. The meristematic cells, that's the growing cells. Okay, the older cells will have more cellulose, but the meristematic cells, which are the growing cells and the at the tip and all, you will find. So these cells involve mostly primary cell wall. Okay, so other points you can ignore. These things you should definitely remember. Okay, and this is like basic point, like cellulose microfilaments is the unit of cell wall organization. Next is secondary cell wall. Okay, it is present between primary cell wall and the plasma lemma. I showed you how it is. Okay, the most uh, whatever the part is inside, that is the plasma lemma. Then you have the secondary, primary, and then in the middle lamella. Okay, this is the last to be deposited and is mainly completely made of cellulose. Mainly it is made of cellulose, but here you see only 30% cellulose. Here it is completely mainly cellulose. Okay, so these things you should remember. Next, we study about the plasma lemma, or it is also called plasma membrane. Okay, what it is made up of, what it is made up of, what is its function? That's what you should majorly know. Like it's made up of 40% protein and 60% lipid. Like the major phospholipids, you should remember, like protein and lipids, 60% is lipids. That's why like phospholipids like lecithin, cephalin are present. Okay. And how these are organized, we have different theories, which we will discuss in very, very brief. Okay. And the chief function, just tell one liner, that much only space you have, not more than that, to regulate the movement of various molecules into and out of the cytoplasm. Okay. To movement, for the movement between the components, we have this plasma lemma, that is the unit of communication. Okay. To regulate the movement of various molecules into and out of the cytoplasm. Movement between and inside and out of the cytoplasm is done by plasma membrane, which is very important and key point which you should remember. So we have two models. Okay. um, Not necessarily you need to remember, just for your understanding. Like you see the protein, and I told you phospholipids are there. So this is the proteins like this. Okay, and the phospholipids are in this pattern. Okay, the hydrophobic zone, and we have the hydrophilic zone. Here we have two layers, one layer here, one layer here of this. Okay, this is lipid bilayer model. Then this is the most accepted, like fluid mosaic model, wherein we say that the proteins are embedded, okay, in the phospholipid bilayer. Okay, proteins are embedded within the bilayer. This is the phospholipids, and these are the proteins. Proteins are just embedded. So these are two theories. Okay, not just remember the theory and one point. That's it. Then about the cytoplasm. Okay, everything surrounded by the plasma lemma except the nucleus is called the cytoplasm. Okay, and cytoplasm minus all organelles, like all the organelles, is called hyaloplasm. Okay, and we have many organelles. You all know. We will discuss them in the next slides. Okay. So what is it? Next, we will be studying about endoplasmic reticulum. This is one of the most important organella. Okay. So we have extensive network of membrane enclosed spaces. See here, it has a more surface area. You can see how this endoplasmic reticulum is there. It has a very high amount of surface area by which it functions. You can see here. Okay, extensive membrane. Next, what does it contain? Okay, it contains the vesicles and the cisterns. Okay, these are the components. And these are not found in RBCs or undifferentiated cells of prokaryotes. It's only present in eukaryote. Okay, it's only present in eukaryote. And another important function is that it is connected with both plasma lemma and the nuclear envelope. Okay, here you have the nuclear envelope. It is connected in this manner with the nucleus, okay, nuclear membrane, and also the plasma lemma, which I told you, it is the outside layer. Okay, the first layer, plasma lemma. Okay, it is connected with these both because it is it is having a network. Okay. Next, it is like originated from the outer membrane of the nuclear envelope. It is about the origin of this endoplasmic reticulum. But more than all these things, you should remember this. Rough endoplasmic reticulum is the one which helps in protein synthesis. This you should never forget. And smooth endoplasmic reticulum. Okay, this is involved in hormone and lipid synthesis. These two you should remember. Other things, okay. Okay, first is rough endoplasmic reticulum works on the on the function of protein synthesis, and smooth endoplasmic reticulum on hormone and lipid synthesis. And make this diagram. That will be sufficient. And the other major functions are like it also provides the mechanical support to the cell. Okay. And exchange of molecules through osmosis, diffusion, and active transport also happens through endoplasmic reticulum. Okay, exchange of molecules, as you know, osmosis and all these things happen. And also, I told you it provides increased surface area because of its network. It provides increased surface area for enzymatic action. Okay, for the enzymes to act, and also forms new nuclear envelope at telophase. This point is not required for you right now because we need to explain a lot to tell that. And the most important is it is the transporting system to different organelles. This you should remember. Okay, it provides channel of transport. Endoplasmic reticulum provides channel of transport, and remember this: rough endoplasmic reticulum, smooth endoplasmic reticulum. Okay, and also it acts as a mechanical support to the cell. Okay, and also another last important point is like endoplasmic reticulum provides passage for mRNA from nucleus to rough endoplasmic reticulum. Okay, so the mRNA which is produced, okay, in the nucleus, and it has to be transported, and it is like controlled by the endoplasmic reticulum. That's why endoplasmic reticulum provides a controlled passage for mRNA from nucleus to rough endoplasmic reticulum. As I told you, rough endoplasmic reticulum is with protein synthesis. So other important organelle is ribosomes. Okay, so I'm telling you key points which you should remember. This is not a whole course. This is what I want you to cover all these things in a very short manner, that any question it comes, this much point is enough for you to write in 3 pages and get maximum marks. Ribosomes. So what are ribosomes made up of? They are made up of RNA and protein. These things you should remember. And we have the 70S ribosome. If 70S is there, then this is present usually in prokaryotes. Okay, we have 80S in eukaryotes. Okay, so this is divided into 30S and 50S. Okay, these details no need to remember. Okay, so these are the units: 50S and 30S. 70S has the 2 units, 50S and 70S. Okay, so it is composed of RNA and protein. The major function of ribosomes is the protein synthesis. Okay, because what happens in case of rough endoplasmic reticulum, why it is involved in protein synthesis? Because it has the presence of ribosomes. Okay, understood. That's why it is involved in protein synthesis. And also, it can support protein synthesis along with the attached to ribosomes, which I told you. The other next important organelle which you should remember is Golgi bodies. Okay, so how is Golgi bodies formed? Like we have 2 to 7 flat cisterns. Okay, cisternae, you can see here cisternae. Okay, they are stacked to close to each other in this manner. The origin of Golgi complex is from endoplasmic reticulum. Okay, but remember now the function, major thing. Packaging plants of the cell. This is called the packaging house. Okay, packaging house or the packaging plants of the cell. What happens here is protein synthesized in endoplasmic reticulum. Okay, you know, I told you rough endoplasmic reticulum, there is protein synthesis because there is presence of ribosomes, and that is transported to Golgi complex where they are stored, often modified and packed into vesicles and cut them off. This is the key point. Means what happens, the proteins produced in the endoplasmic reticulum are transported to the Golgi complex. Okay, they are transported to the Golgi complex where they are stored. Okay, often modified. This is the storage house. All these the proteins are stored, often modified, and packed into vesicles and cut off from them. Okay, means the next slide. Okay, what happens is the here we have the vesicles, and these vesicles will fuse with the food molecules and or the protein molecules and packed, and then they cut off and move to different organelles. Okay, so this is what happens here. So remember, they are the stored, often modified, and packed into vesicles and cut them off. They cut them off, and then they go to the place it has to go. Okay. So this newly formed vesicles, I told you, they fuse with the plasma lemma and transfer to outside cells. So they will just break. Okay, this will break, and then they will go and fuse with the plasma lemma. Okay, plasma lemma, which I told you on the cell wall, plasma lemma, plasma membrane or plasma lemma. So uh, then they are transmitted. So these vesicles are the key. Okay, newly formed vesicles, you should remember. Then the next most important is lysosomes. Okay, this is as we call the acid digestion, as we have the acid phosphate and DNAs and the RNAs. It is mainly involved in having the hydrolytic enzymes. Okay, and it also fuses with the food molecules. So the fusion of primary lysosomes with the food molecules through phagocytosis or pinocytosis. Okay, here you can see. Here we have the molecules like the food molecules or some any foreign body. That is what is the use of lysosomes. Okay. So what they do, they engulf the food molecule. Here you can see they engulf the food molecule. Here you can see how they engulf the food molecule, and they then the lysosomes acts on them and they break them, and this is called exocytosis and eliminate them. Okay, this is like it has a digestive enzymes. Okay, the lysosomes. So here, what is the function is that lysis of food materials or microorganisms engulfed in the cell by autolysis of the cell if required. Okay, so what it does, first you should remember it has acid phosphate, acid DNAs, and acid RNAs that are having the hydrolytic uh, power. Okay, and then the molecules, then they fuse with the lysosomes, and then these are broken away. Okay, so these used in like some microorganism attack or some food materials also they are lysed. This is not required. So next is vacuoles. What are vacuoles? Vacuoles are the materials inside the vacuole. Okay, material inside the vacuole is called cell sap. Okay, whatever is inside is called cell sap. And the outermost layer of this, okay, so the outermost layer of this is called tonoplast of vacuole is called tonoplast. Okay, this vacuole provides the physical support and also the rigidity to the cell. Okay, and here we have many different components also. So all these things we will discuss in the next coming slides, and also like the ion balance. Okay, water balance are all controlled by the vacuoles because it provides the required concentration. Okay, for all the cells to be there and for the movement of molecules. Next, the most important is we are discussing is about mitochondria. Okay, so what about mitochondria? So it arises by the fusion of pre-existing mitochondria. The pre-existing mitochondria leads to this the other mitochondria. Okay, so it is composed of 70% protein, 25% lipids, and the internal structures you can see here, cristae. These are the internal ones. These are called the cristae. Okay, the inner membranes are called the cristae. The outer membrane contains the protein porine, okay, which are involved in molecular movement. See here, this is the one of the most important function, cristae. Okay, granules are there, then ribosomes are there, matrix is there in the inside, DNA is there. Here you can see the DNA molecules, the inner and outer membrane, all these things are there. Just remember it. Okay, so what is its use? Okay, this complete detail we'll discuss in physiology. See, in the electron transport chain, you would have seen. Okay, in case of electron transport chain, so here it is the mitochondrial matrix. Okay, here we have the mitochondrial matrix. Clearly here you can see now. Here we have the mitochondrial matrix. Okay, and this is the The diagram which is showing is the inner mitochondrial membrane. So you know how in the intermembranes, and this is the intermembrane space, on how the electron transport chain occurs. Okay, it is useful for the oxidative phosphorylation, okay, which is the basis for the production of ATPs. Okay, and also here is ATP synthesis in the matrix. Okay, matrix of the mitochondria. So these are the two important functions, and how this works is there in our physiology portion. So that will this how it works, that we'll discuss in the physiology. So here we should remember it is involved in electron transport chain. Okay, and which leads to oxidative phosphorylation and production of ATP in the matrix. These are the key points in case of mitochondria, or it is also called like the powerhouse of the cell. Okay. So in plastids also you have different types like leucoplast, which is colorless, and chromoplast, okay, which has different other pigments like phycocyanin, keratin, and xanthophyll. So in case of chloroplast also, just remember the structure. We have outer membrane, then a granum, okay, and at the individual level, thylakoid membrane is there, then lumen is there, and the stroma lamella. Okay, they are also involved in different procedures that we'll discuss in the coming slides. Okay, we also have a circular DNA because only in case of mitochondria and chloroplast we have DNA. So these things you should remember, and the major functions. So here what happens is the light reaction, which also these topics we will read in the physiology portion. Okay, but if they ask in genetics, you just need to mention. You don't have space to write what in detail. Only this much, whatever content I'm giving. If you write this, there is more than sufficient. So what happens is it is light dependent phosphorylation. It takes place inside the chloroplast, and light reaction happens in grana. Okay, and dark reaction happens in stroma. Stroma means the inside one. Okay, this is the thylakoid or the grana. Okay, wherein the light reaction happens. Okay, you know that light reaction, then H2O, then oxygen is released, and from the Calvin cycle we get the NAD plus and ADP. Then there is production of ATP, NADPH, and which acts as the source for the Calvin cycle. And finally, there is production of sugar. Okay, or this is the basis of photosynthesis. Okay, this is the basis of photosynthesis. Also, you would remember about how the PS2 and PS1 acts in case of non-cyclic photophosphorylation for the production of sugars. Okay, by phosphorylation, and lead to the production of sugar, which is the basis for photosynthesis. Okay, so this diagram also you should remember. Okay, light reaction, then Kelvin cycle. Okay, how it moves from ADP is given away from Kelvin cycle ADP, and then from light reaction we get ATP and NADPH. Then we produce sugar. So this complete detail we will discuss in physiology, because this is not class for discussing all these things in detail. Just I'm telling you points. So next is about nucleus. Okay, so till here we have discussed about all these aspects. So for watching the full video, okay, for watching the full video, you can find in the description. You can log into the channel